Okay, so welcome back to this next video in which we are discussing cyclic AMP signaling. Okay, right. So we're currently in the process of discussing how calcium ions can regulate adenylyl cyclases. So we've just been looking at how calcium acting through calcium calmodulin complexes can stimulate adenylyl cyclase 1, 3 and 8, the group 1 adenylyl cyclases, okay? And we looked in detail at where the calcium calmodulin complexes bind to the adenylyl cyclase 1 and adenylyl cyclase 8 enzymes. Those kind of details aren't yet known for adenylyl cyclase 3, but we do believe that calcium calmodulin complexes regulate adenylyl cyclase 3 enzymes. Okay, right, so going back to our classification of the 9 plasma membrane adenylyl cyclases into these 4 groups, okay, we've now covered the calcium-stimulated group 1 adenylyl cyclases. We understand how they are now stimulated by calcium. We've covered the group 2 G-beta gamma stimulated adenylyl cyclases. We understand how those are stimulated or at least potentiated by G-beta gamma subunits. Okay, we've done group 4 adenylyl cyclases long ago. We've discussed how the group 3 adenylyl cyclases are inhibited by G-alpha I/0 subunits, but we haven't yet discussed how calcium inhibits the group 3 adenylyl cyclases, so it's that I now want to turn my attention to. Okay, right. So the first thing to say is that all plasma membrane adenylyl cyclase enzymes are inhibited by calcium if you put the calcium concentration high enough, okay? But you have to raise it for most of them to non-physiological concentrations. It's only the adenylyl cyclase 5 and adenylyl cyclase 6 which are inhibited by physiological calcium concentrations. So let me explain why all adenylyl cyclase enzymes are inhibited by calcium if you raise the calcium high enough. So let me start by drawing another picture of an adenylyl cyclase enzyme. So here we have the amino terminus of the adenylyl cyclase enzyme. Here we have transmembrane domain 1 here, that cluster of 6 membrane spanning alpha helices. So this is TMD1. And I'll just colour in the membrane spanning alpha helices here in vivid purple. Okay, then after transmembrane domain 1, what you have then is the C1a domain, which we'll have here. Okay, then you have the C1b domain, this portion here. Then the next cluster of 6 membrane spanning alpha helices, which is here, which is transmembrane domain 2. And I'll colour in those 6 membrane spanning alpha helices of TMD2 in vivid purple. Okay, and then after TMD2 what you have then is the C2a domain, which will be dimerized with C1a and making the actual catalytic domain of our adenylyl cyclase enzyme. Then you have the C2b domain and then the C-terminus. Okay, so here is the catalytic domain of our adenylyl cyclase enzyme. Now basically all adenylyl cyclase enzymes have a really important divalent cation at the center of their catalytic site. So I'll draw this here. Okay, and this divalent cation is a magnesium cation. Okay, which is in the same group of the periodic table as calcium. It's the smaller one, the one that's above calcium. Okay, so here is magnesium. Right, now this magnesium is absolutely essential for the catalysis of the reaction, basically, the conversion of ATP into cyclic AMP and pyrophosphate. Okay, indeed it coordinates, it's involved in coordinating the ATP and bringing it in here for the reaction to actually occur. Now, basically, when you put the adenylyl cyclase in a very high concentration of calcium, the calcium ends up displacing the magnesium from this site, and you get a calcium ion in there instead of magnesium. Now, why does that cause a problem? You'd think that the calcium could still coordinate the ATP. After all, what brings the ATP in is the double positive charge. Okay, calcium still has the double positive charge. So why does the calcium not work? Well, the problem is calcium is bigger than magnesium and can actually bind to a larger number of things. So let me show this. Okay, so if we have the magnesium here, the calcium is a bigger ion. So this is the calcium here. And basically, this means that calcium can have more things coordinated around it than magnesium. Magnesium actually has a valency of 6. It can have one thing here, one thing here, one thing binding here, one thing binding here, one thing binding out the front, and one thing binding at the back. Okay, so the valency of magnesium, I was about to say the magnesium of valency. The valency of magnesium is 6. Calcium being bigger has a bigger valency. It can bind to 8 things, okay, in a cube effectively around it. Okay, so 1, 2, 3, 4, and then on the bottom here, 1, 2, 3, 4, forming a cube of things that are bound to the calcium ion, like so. Okay, so it has a valency of 8 rather than 6, so it can bind to 8 different things at the same time, basically. Right, now why is this bad? Well, basically, this means that the pyrophosphate that you generate in the conversion of ATP to cyclic AMP and pyrophosphate doesn't actually get extruded from the active site once it's been made. So when you've got the magnesium here, the pyrophosphate, that molecule of 2 phosphate groups bound together here, gets extruded once the reaction has actually occurred. It gets chucked out. There's nowhere for it to bind. However, when you've got a calcium in there, you've got 2 extra binding sites, and basically the pyrophosphate can bind to these 2 extra binding sites. Here's the pyrophosphate crudely shown here binding in those 2 extra binding sites. So with calcium having this additional 2 binding sites, basically, it has this valency of 8, you get the pyrophosphate stuck in there, basically, and it doesn't come out. And now the enzyme is effectively inhibited because now it can't bring in another molecule of ATP until that pyrophosphate goes out, okay, and the pyrophosphate isn't going to come out, basically. So that's why calcium ends up inhibiting the adenylyl cyclase enzymes. It ends up replacing the magnesium ion, and it can do the job. It can bring the ATP in. The problem is that once the reaction has actually been catalyzed, the pyrophosphate, which is supposed to leave, doesn't leave. It stays in there, and that is what then inhibits the adenylyl cyclase enzyme. Okay, so as I say, this happens for all 9 plasma membrane adenylyl cyclases if you put them in high enough calcium concentration. However, usually for most of them, that calcium concentration that you have to put them in is non-physiological. You never ever get calcium concentrations like that within the cell. Okay? The only 2 adenylyl cyclases which can actually be inhibited at physiological calcium concentrations are adenylyl cyclase 5 and adenylyl cyclase 6. So something about this binding site for the magnesium ion in adenylyl cyclase 5 and adenylyl cyclase 6 means that they have a higher affinity for binding calcium than the other adenylyl cyclases do. Okay, so they will replace the magnesium with calcium at a lower calcium concentration than would the other 7 plasma membrane adenylyl cyclases. Okay, so this means that physiological calcium concentrations can therefore inhibit these adenylyl cyclase 5 and adenylyl cyclase 6 enzymes in exactly this mechanism that I've just described to you. Okay, right, so that's how calcium can inhibit the adenylyl cyclase 5 and the adenylyl cyclase 6 enzymes. Okay, now what I want to talk to you about is a clever concept here, another clever concept, okay, because these adenylyl cyclases, which I've told you about, that are regulated by calcium, okay, so the adenylyl cyclase 1, 3, and 8, which is stimulated by calcium, and the adenylyl cyclase 5 and 6, which are inhibited by calcium. The calcium needs to have come from a certain source, basically. So let me just overview calcium signaling for you. Okay, so if you want to raise calcium concentration in the cytoplasm, there are loads of different ways that you can do it. So if we've got our cell here, calcium concentration in the cytoplasm is usually very, very low. It's usually around 100 nanomolar, so a very, very small concentration of calcium in the cytoplasm. Contrast that to the extracellular calcium concentration, which is around 1.5 millimolar. So there's a 15,000-fold gradient of calcium across the cell membrane. Okay, so if you want to raise the concentration of calcium in the cytoplasm, there are two major ways that you can do this. Either you can allow calcium in from the extracellular fluid into the cytoplasm, or you can release calcium from an intracellular organelle. Okay, and there is an intracellular organelle which is capable of storing calcium called the endoplasmic reticulum. So the endoplasmic reticulum has a higher store of calcium in it than the rest of the cytoplasm. So the ER is a major intracellular calcium store. So this is the major intracellular calcium store. There are other intracellular calcium stores beyond the ER, but this is the major one. Okay, right. So that means that there are two ways of getting calcium concentration in the cytoplasm up. Either you can let it in from the extracellular fluid, or you can release it from the ER. Okay, now, how can you let it in from the extracellular fluid? Well, you could, for instance, have voltage-gated calcium channels, okay, such as the T-type voltage-gated calcium channels that we saw in the cyanoatrial node cells. Okay, but there are many other types of voltage-gated calcium channels as well. So this is a voltage-gated calcium channel which would open in response to depolarization, usually of the electrical potential difference across the cell membrane, and would allow calcium into the cell. Okay, alternatively, you can release it from the ER. Now, how do you release it from the ER? Well, you activate G-protein coupled receptors on the cell surface membrane, which then result in the generation of IP3, which can then open IP3 receptors in the ER membrane. So let me just briefly describe to you the GQ cascade. Okay, so once again I'll draw a G-protein coupled receptor here. So now we're looking at another heterotrimeric G-protein beyond the GS heterotrimeric G-proteins, beyond the GI/O heterotrimeric G-proteins. Now we're looking at GQ/11 heterotrimeric G-proteins. So this now is going to be a GQ/11 coupled G-protein coupled receptor. Okay, and some major examples of GQ/11 coupled G-protein coupled receptors are alpha-1 adrenoreceptors and also M3 muscarinic acetylcholine receptors. Okay, right. So basically, when this GQ/11 coupled G-protein coupled receptor is activated by its ligand, it will go into a conformation where it's capable of activating GQ/11 heterotrimeric G-proteins. Okay, now I won't bother drawing the GQ/11 heterotrimeric G-protein again, I'll just represent the full thing now as a blob. Okay, so this represents the alpha, the beta, and the gamma subunit. Okay, and basically GQ/11 heterotrimeric G-proteins are heterotrimeric G-proteins where the alpha subunit is either alpha Q, which is the most famous one, or it's alpha 11. Now, alpha 11 is less famous than alpha Q, but again, it's almost identical in structure to alpha Q, and it does the same thing as alpha Q. Okay, so GQ/11 heterotrimeric G-proteins are just heterotrimeric G-proteins where the alpha subunit is alpha Q/11. So when you activate the GQ/11 coupled G-protein coupled receptor, it's going to now activate our GQ/11 heterotrimeric G-protein. You're therefore going to generate the on-state alpha Q/11 alpha subunit. So I'll draw this now. So here is my alpha Q/11 alpha subunit with GTP now bound to it here. Whoops, not GTR, GTP. Okay. And what this is then going to do is it's going to activate enzymes called phospholipase C beta enzymes. Okay, now there are loads of phospholipase C enzymes. A family of phospholipase C enzymes are the phospholipase C beta enzymes, and these are all the time moving between being attached to the underside phospholipid bilayer and being in the cytoplasm. Okay, so this PLC beta here, this stands for phospholipase C beta. Okay, and there are 4 different genes that code for phospholipase C beta enzymes. There's phospholipase C beta 1, phospholipase C beta 2, phospholipase C beta 3, and phospholipase C beta 4. Okay, but for our purposes they all do exactly the same thing, so we'll just abbreviate them all down to PLC beta. So PLC beta is this enzyme that is all the time relocating around the cell. Okay, sometimes it will be floating in the cytoplasm, other times it will be attached to the underside of the plasma membrane. So it's capable of relocating itself. However, it's never going to be active until the alpha Q/11 subunit activates it. Okay, so just attaching to the underside of the phospholipid bilayer is not good enough to activate it. It just relocates it. Okay, now the alpha Q/11 subunits with GTP bound can then activate the phospholipase C beta enzymes, which are currently attached to the underside of the plasma membrane. So when phospholipase C beta enzymes are attached to the underside of the phospholipid bilayer, they can then be activated by on-state alpha Q/11 proteins. And once they've then been activated, they will start breaking down a membrane phospholipid called PIP2, standing for phosphatidylinositol 4,5-bisphosphate. So I suppose I probably should write its name out in full. So this is phosphatidylinositol, that's the PI, okay? And then it's 4,5-bisphosphate. Now this is a normal component of the plasma membrane, okay? It's a phospholipid. It's not an awfully common phospholipid. It's quite a rare phospholipid in the plasma membrane, but it is a normal component. So you do have phosphatidylinositol 4,5-bisphosphate molecules normally within the plasma membrane. Okay, then what's going to happen is it's going to break down the phosphatidylinositol 4,5-bisphosphate molecules in the cell membrane to IP3 and diacylglycerol. Okay, the diacylglycerol will remain attached in the phospholipid bilayer, so it will remain in the phospholipid bilayer, so it's a diacylglycerol there, that's the DAG. Meanwhile, the IP3, which is the inositol 1,4,5-trisphosphate, this will drop down into the cytoplasm, so this doesn't remain attached to the underside of the phospholipid bilayer anymore. It was just attached because it was attached to the diacylglycerol. Okay, now what's going to happen is it's going to drop into the cytoplasm. So this is inositol 1,4,5-trisphosphate. So the I is for inositol, and then the P is for phosphate, and then because you've got 3 phosphates, it's IP3. Okay, right. Then what's going to happen is this IP3 that drops into the cytoplasm is then going to act on receptors in the ER membrane called IP3 receptors. Okay, and IP3 then causes the IP3 receptors to open, and then you can have the release of calcium from the ER. Okay, so that's another way that you can get calcium signals occurring in the cytoplasm. Okay, now the final way I want to talk about how you can get calcium coming into the cytoplasm involves what's called store-operated calcium entry. Okay, and this is going to be one of the ways in which you can get calcium activating the adenylyl cyclase 1, 3, and 8, and also inhibiting the adenylyl cyclase 5 and 6. So that's why I'm going to bring this up. Okay, so we're now going to talk about store-operated calcium entry, okay, which often is abbreviated down to SOCE for short. Okay, right. So what is store-operated calcium entry? Well, basically, when you get calcium being released from the ER like this through IP3 receptors, the calcium level in the ER goes down. Okay, and what that then triggers is it triggers the activation of a cascade which aims to refill the ER back up. Okay, so let me explain this. So basically there are proteins in the ER whose job it is to measure the amount of calcium in the ER, and if the amount of calcium in the ER is too low, they are then going to activate a pathway which is going to result in the refilling of the ER, basically. Okay, and these proteins are called STIM1 proteins. Okay, so this protein is called STIM1, and STIM1, which I'll colour in vivid purple here, stands for stromal interaction molecule 1. Okay, so I'll put this down here. So STIM1 stands for stromal interaction molecule 1. So the ST is for stromal, the I is for interaction, okay, and then the M is for molecule 1. So the stromal interaction molecule 1. Now, normally, when calcium level in the ER is normal, then the stim1 does nothing, okay? However, when calcium goes down within the ER, what starts to happen is you get stim1 proteins aggregating together in the ER membrane, okay, like so. So we now get these stim1 aggregates here, so I'll colour these in vivid purple. So these are loads of stim1 proteins aggregating together, and these stim1 aggregates are then capable of activating calcium channels that are in the plasma membrane here. Okay, and these calcium channels are known as the calcium release activated calcium channels, or the CREB channels. Okay, so these are the CREB channels, which stands for calcium, that's the first C. Okay, and then it's release activated, so the R is for release, the A is for activated, and then the second C is for calcium again. Okay, so it's the calcium released activated calcium channel, or the CREB channel. Okay, right, so I'll colour in the CREB channel in orange here. Okay, so the stim1 aggregate opens the CREB channel. You get calcium coming in from the extracellular fluid, and then the idea is that there is a pump on the ER membrane which can then pump calcium back into the ER, basically. And that pump, which I'll colour in yellow, is known as the CERCA. Okay, so in yellow there, that's the CERCA pump, which is the sarco/endoplasmic reticulum calcium ATPase. So basically, this pathway is to refill the ER lumen with calcium once it's been released into the cytoplasm, and this pathway is called store-operated calcium entry. Okay, so I'll just write out what CERCA stands for. So CERCA is a special pump for pumping calcium back into the ER, and that's called the sarco/endoplasmic. Okay, so that's the SE, and then it's reticulum, which is the R, and then it's calcium ATPase. Okay, and this uses ATP hydrolysis to provide the energy that it needs to pump calcium into the ER. Specifically, it pumps 2 calciums into the ER for each ATP molecule it hydrolyzes, and it also actually takes 3 protons out of the ER. So if I draw it in a little bit more detail here, and this is the reaction it does. So this is the ER membrane here. It will move 2 calcium ions into the ER lumen, okay? Meanwhile, it will take out 3 protons from the ER into the cytoplasm, and every time it does that, it will hydrolyze a molecule of ATP down to ADP and inorganic phosphate. Okay, so that's how the CERCA returns calcium back into the ER. Okay, so that's a whiz tour of calcium signaling then, and these different ways that you can get calcium going up within the cytoplasm, because obviously when you do activate these CREB channels, the calcium first goes into the cytoplasm before it then returns into the ER, so that's another way of getting calcium to rise in the cytoplasm. Okay, now, basically it turns out that all of these adenylyl cyclases, which are regulated by calcium, okay, both the group 1 adenylyl cyclases, which are activated by calcium, and the group 3 adenylyl cyclases, which are inhibited by calcium, they're only affected by calcium that comes from specific locations. Okay, and those locations are the voltage-gated calcium channels and the calcium release activated calcium channels. Okay, you cannot get the adenylyl cyclases being affected by calcium that's being just released from the IP3 receptors. Okay, so what I am saying here is calcium coming from the ER out of IP3 receptors is not capable of regulating any of the adenylyl cyclases that I've told you about. Okay, calcium from voltage-gated calcium channels is capable of activating and inhibiting the adenylyl cyclases that I've told you about. Okay, and calcium coming from the extracellular fluid through calcium release activated calcium channels is also capable of regulating the adenylyl cyclases. Okay, so why? Why is that the case? Well, basically, we think the answer again lies in the fact that calcium signals, like cyclic AMP signals, are not global. Okay, so we do not believe anymore that calcium signals are necessarily global. Okay, it's not the case that whenever you get a calcium signal, calcium goes up everywhere in the cytoplasm. Instead, we think that there are probably calcium signaling microdomains, just like there are cyclic AMP signaling microdomains. And in those microdomains, you'll have specific ways that can bring calcium into the cytoplasm, along with specific targets for that means by which you can bring calcium into the cytoplasm. And indeed, what we've started to find is that all of these adenylyl cyclases that I've told you about, adenylyl cyclase 1, 3, 8, 5, and 6, the ones that are regulated by calcium, you can find them attached to voltage-gated calcium channels and also attached to crack channels. Okay, so it seems as though they are in little microdomains with these means for increasing cytoplasmic calcium, and therefore it's likely that the calcium signals that you're generating here are localized calcium signals, and that they're having effects on those adenylyl cyclases in this little microdomain of calcium signaling, basically. And that's why IP3 receptor released calcium isn't capable of activating these adenylyl cyclases or inhibiting these adenylyl cyclases in the case of the group 3 adenylyl cyclases, because they're not in the same microdomain as the IP3 receptors, basically. Okay, right. So that now concludes our discussion of cyclic AMP signaling.